ahead and get started to keep everything on track tonight. So enjoy everybody for joining on tonight for the last North Carolina Small Ruminant Improvement webinar for this winter series. Tonight we have two guests with us, Dr. Jake Thorne. He is the assistant professor and extension sheep and goat specialist at Texas A&M, and I think he's stationed in San Angelo, Texas, out there in West Texas. And then we second we'll have Dr. Lee Wright. He is superintendent of Southwest Agriculture Research and Extension Center and the senior research associate at Virginia Tech University. So we're pleased to have them on tonight. And tonight's topic of conversation is going to be genetic selection. Just a couple of housekeeping rules. We're going to do the two presentations this evening. We'll have a session of Q&A after that. And if you don't mind, put in the chat where you're from. If you're from North Carolina, put in what town or county you're in. And if you're joining us from across the state or anywhere else in the nation or countries, put that in as well. But we'll go ahead and get started with Dr. Thorne. And you should have access to share your screen there. And I will turn it over to you.
Okay. Can you still see that?
Yep, well, you're good.
Perfect. All right. Well, thanks, Adam and Andrew and the whole crew for inviting me to come and speak this evening. It really is an honor. I'm obviously going to, from a little bit different area than the rest of y'all. And so maybe some of what I'm going to talk about is maybe not directly the same environment that a lot of you are in. But as I go through genetic selection of sheep and goats, kind of how we prefer, how we approach it here in Texas, I guess one of the things that I really would like you to take away is how important that environmental component is, and selecting animals that are appropriate for your type of production setting or your type of environment. That's really critical as we kind of approach this to be economically successful. So I'd like to start, just take a few minutes here at the front end, just to give you guys a little bit of a background about myself and kind of where we're at out in West Texas. As Adam said, I am stationed at the Texas A&M AgriLife Research and Extension Center that's in San Angelo. And so that's kind of west central Texas. And really the mission of our research center is to provide small ruminant ranching solutions. And so to accomplish that, we've got Dorper and Rambouillet sheep, kind of a Spanish boar cross, and Angora goats. Historically, Texas was absolutely a fiber producing state. The vast majority of the sheep were Rambouillet sheep. The vast majority of the goats were Angora goats. But I'd say over the last 30 years or so, as that gap in the value of the kid or lamb versus the value of the fiber that can be shorn off those animals really widened, there was really a transition, or starting the transition towards Dorper sheep specifically, hair sheep, but predominantly Dorpers, and then also meat goat type animals. So at the same time, markets for those types of offspring that really prioritized, really valued slaughtered lambs and kids that were a little lighter weight really started to emerge. And so today, I don't know if we know exactly, but I would say that probably close to 70 or 75% of the sheep in the state of Texas are now Dorper or Dorper influenced of some kind. And then it's probably over 90% are meat goats. We have pretty active genetic and parasite and grazing research programs, and along with a pretty active livestock guardian dog extension and research program. Predation is a huge issue in the state of Texas. I'll touch on that a little bit more later. But getting guardian dogs out there is important to our stakeholders and our producers, and important to our station. Without them, our research would be, we'd be in a lot of trouble, absolutely. The goal and then the way that I like to describe it is we're really trying to conduct intensive research in an extensive setting. So I mentioned earlier that we're trying to provide ranching solutions, and we're trying to conduct our research and have these herds and flocks and manage them in a situation that is really similar to how small ruminants are produced in our region. And that is a more extensive, hands-off, lower input type of situation, larger pastures, larger scale. And so a lot of what we do, even though it maybe makes it a little bit more challenging in some instances to collect data, we try to really reflect that regional production system. So group mating, pasture lambing and kidding. All of our females are expected to gestate out in the pasture, give birth, raise their offspring out in the pasture away from the barn. We try to provide as little supplementation as needed. Absolutely, we'll use it in certain conditions, certainly during dry periods. But we try our best to make those animals forage and graze and get what nutrition they need from out in the pasture. A little bit more information about our environment here. San Angelo is on the northern side of Edwards Plateau. And this is absolutely a subjective statement here. Edwards Plateau, in my opinion, is good sheep and goat country. That might not be the case for a lot of other people and their perspective, but it's a little bit drier. We get 20 to 25 inches of rainfall annually. When it does come, though, it's during the summer, later spring, early summer. And our region is prone to drought. And so we'll go several years with less than that annual rainfall, and then we might get a year where it's double that rainfall. And what makes that challenging is that we still have a really intense parasite problem. And what that comes from is that the breeds that I mentioned that we run or that are popular in the state of Texas are much more on that susceptible end of parasites when we talk about parasite resistance. And so when we do get years, when we get good rainfall and there actually is forage out there, it's really a goal of mine and my genetics program to make sure that we have animals that can at least withstand that moderate parasite challenge and take advantage of that good forage year. As it stands right now, for a lot of producers, because of the buildup of anthelmintic resistance and whatnot, that when we have our wetter years, they're almost in worse situation than we're in a drier situation. So again, like I mentioned, that's really a goal of my genetics program and what we work on here. We are blessed with more mild winters compared to certainly our northern friends. That does provide quite a bit of cool season grasses and quote unquote weeds that allow for some good quality forage through the later fall and early winter for our livestock. And it really promotes our ability to have small grain production. And so what that presents an opportunity to our Texas producers, we maybe as a state don't take advantage of this quite as much as we should, is really fall lambing, weaning, and marking offspring during the low supply time of the year. And so the figure that I've got here, this is from San Angelo, the livestock auction here in San Angelo, but it's really reflective of kind of the national industry, is that we have such a high supply of lambs that go to town during the summer and early fall. And consequently, there's price depression during that time. And at the same time, during kind of starting about Thanksgiving and into Christmas and through March, prices are much, much higher. And so those flocks are producers that can take advantage of kind of that aseasonal time period, breeding in the spring, lambing in the fall, weaning during that winter and marking those offspring that time period can really take advantage of that increased price. And so when we think about some genetics and how to select animals that fit that model, having that endpoint in mind is really critical. Okay, I'm going to take a step back here and kind of focus more on genetics. The history of genetic selection in Texas is long and vast. Kind of the one thing that I really want to point out here is that for a long time, our research center hosted a performance Rambouillet ram test. Started in 1949 and went for almost 70 years. And you might be familiar with that format, but essentially consigners would bring rams in. They would stay at our station for about 6 months or so. We would put them in a dry lot. They would be on full feed. We'd measure their growth. We'd measure their wool quality. There'd be a pretty intensive visual appraisal that includes structure, that would include skin folds, body, various composition. And there's absolutely no doubt that over the course of this test, that those traits were improved in the Rambouillet breed. So, specifically in our region, but across the country, there's been other central performance Rambouillet tests, and that model has really improved that breed for those traits: improved growth, improved wool quality, improved appearance. But at the same time, we kind of missed out on measuring some traits and maybe took it a little too far by ignoring some things. And that included reproduction. So I mentioned that it was just a ram test. They were just evaluated for about 6 months. There wasn't anything reproductively speaking that data was collected on. We didn't capture anything in the form of parasite resistance. And maybe the biggest issue was that these rams were put in a dry lot and put on full feed, but then when they went home, and really for the rest of their life, and the offspring that they produced were all expected to survive in not that type of situation. So we weren't really evaluating the fitness of these bucks to the environment that they were going to be involved in or be ran in once they went back to the ranch. So what that led to was kind of a challenging situation. So I got a picture here on the left side of the slide that I honestly just went out in one of our pastures here a few days ago and snapped. And you'll notice, I mean, it looks pretty dry. There's not a lot out there to eat. Forage quality is probably a little lower. It is winter time, and we have for however long that ram test ran, pretty much prioritized that as the selection model in a Rambouillet flock. And what that led to was animals that grew fast, but were also a little bit larger in frame. They needed that extra nutrition to stay in proper condition. And so it's not abnormal for some of our Rambouillet sheep to get in the situation like I've got pictured here on the right. They stay a little too thin. They're just a little bit too large frame to fit the country. And so that is, again, a goal as we kind of shifted our focus over the last 5 or 10 years, trying to downsize those animals and really prioritize their ability to survive in a pasture setting. At the same time, I've shown you here maybe a little bit more drier visual, but like I said, and like I mentioned earlier in the presentation, when we do get summer rainfall, we want our animals to be able to take advantage of that and not succumb to parasites at the first sign or at the first instance where those parasites show up in the pasture. Okay, kind of focusing in on reproduction. And this is maybe a little bit embarrassing to show, but it's truth and fact, and I wanted to present it tonight. This is from the latest USDA NASS report that just came out the end of January. And this presents the lamb crops for each of the various states in the U.S. And if you look down here at Texas, I'll see if I can get the laser here. I mean, our state lamb crop is 79%, which is not very good, or not really even close to very good. And now I'll admit that does come with a bit of a disclaimer. I'm not sure if you can see the bottom here. It looks like this panel of controls is a little bit in the way, but lamb crop is defined in the western states and in Texas as the number of lambs that were identified at marking versus the number of ewes that were exposed. And that is a little bit different than some of our eastern states, which be the number of lambs that were born. And what that means is that number is influenced somewhat by neonatal lamb survival. And we absolutely have a predator issue in the state of Texas. Coyotes are really reemerging. We have a growing feral pig problem. There's always birds of prey that help contribute to predation. And so when I look at this 79%, you have to take it a bit with a grain of salt. But at the same time, it's not all on predation. Our sheep and our goats both have just really not been selected enough for more prolific females that are able to not only give birth, but raise those offspring. And we should be up closer to that 130, 150% lamb crop. So we've got a ways to go. Okay, so I put together a few bullet points as to what I kind of view as either some goals or kind of the ideal commercial Texas ewe or nanny. And so I want to go through some of these because, again, I think this really drives our genetic selection program and what we prioritize. And so I want all of the ewes or females in our flocks and herds to make sure and get pregnant every single year. No missed pregnancies, and really like them to breed first cycle. So what that means is that the estrus cycle of a sheep is 16, 17 days, somewhere in there. If we expose them to the rams for breeding season, let's say a 40-day breeding season, really like those females to get bred in the first 17 or first half of that breeding season. I'd like twins 2 out of every 3 years and 6 lifetime parities. So what goes into that is some longevity, absolutely, in those females, but also some fecundity, some prolificacy, in being able to get bred up early and to have at least twins 4 out of those 6 years. I like those females to wean their body weight in offspring. And so that's certainly going to be more possible for ewes that have twins. If we're looking at 140-pound ewe, if she's got 2 70-pound lambs, she'll be able to reach that benchmark. If she's got just a single, obviously that's probably not going to be likely to happen, but that's still the target goal here. I really like those females to give birth and raise those offspring out on pasture on their own with somewhat minimal supplementation. Obviously, there's going to be some instances or some environmental conditions that are going to require some supplemental feed, but at the same time, the majority of their diet should come from forage or what they're grazing on. For our wool sheep, I'd like them to shear an 8-pound fleece that's at least sub 19 micron. That falls in a pretty high value range there that obviously doesn't apply to our Dorpers. And I really like those ewes to stay in proper body condition. And so kind of thinking about how we would genetically select a female to stay in proper body condition. Kind of the first thing is to really make sure that they don't get too big. And so yearling weight or body size are something that we really select for in moderation. In some cases, like for smaller animals, we would like them to wean heavy and grow fast through that post-weaning time period, but not just continue to grow through that first or second year of their life so they don't get too large. And then, as I mentioned a couple of times, really like them to be able to withstand at least a moderate parasite challenge. I recognize that Dorpers, Rambouillets, Angoras, Boer goats, those are all breeds that are fairly susceptible to barber's bollworm compared to our Katahdin or St. Croix or Caribbean breeds. And so I can't expect that when faced with just a massive worm burden out on pasture, that maybe they're going to be okay with that. But at least a moderate parasite challenge, we want them to be able to withstand. Okay, so how do we get there? We've kind of started with the end in mind. I described quote unquote an ideal ewe. So how do we get our flocks or our herds into that type of situation? So really a good practice that I would recommend to all of you is to make sure and capture performance records. That's kind of step one. And so you have to use technology, or I encourage you to use technology to keep good records. That includes parentage or pedigree, collect body weights, collect fecal leg counts. And that technology aspect of that is just going to maintain the accuracy of that record keep. And so that might be things like using EIDs or using spreadsheets, using certain livestock management programs. There's many out there that, whatever kind of fits your type of production setting. But using that type of technology makes it easier and makes it more likely that you'll go ahead and collect that data, and obviously makes it more accurate. Select your keepers, so your replacement females or your replacement rams from the right type of ewes. So as I mentioned earlier, appropriate size, in good body condition, maintain good body condition throughout the year, are adaptable to the type of situation and environment that you're putting them in. Select lambs that were born and raised as twins. That's a good step. Now, most reproductive traits are pretty lowly heritable. And so what that really means from a practicality standpoint is that if you just select lambs that were born and raised as twins, that you probably won't make really drastic increases in terms of your lamb crop or in terms of those reproductive traits in the short term at least, but you're not necessarily going to go backwards. And then also collecting fecal leg counts, particularly during that weaned period or post-weaning period. So, if we collect an egg count from a lamb that's still on its mother, there's a maternal influence that kind of makes, I don't want to say confounds, but it makes it challenging to look at that fecal leg count from a genetic standpoint. Once those animals get much older than a year or two and are in production and raising their own lambs, again, it becomes pretty challenging to parcel out what's genetic versus maybe some stress or different environmental factors that are causing a fecal leg count to be at a certain level. And so during that post-weaning time period, when everybody is still growing, but there's not much maternal influence, they haven't entered into production yet, that's really the ideal time to measure something like fecal leg count. But with all that said, the best practice is to take those raw records, those raw data, and get them into the National Sheep Improvement Program to generate estimated breeding values. And so if you're not familiar with NSIP and EBVs, it's a quantitative genetics program, and the output of it are genetic predictors for a whole suite of traits, including many of the ones that I've described and some others that I haven't so far. Really, those calculations are based off of performance records, but also heritabilities that vary from trait to trait. They include or are based off of how many records are into that program. Generally, more data, more information. The result of that is more accurate EBVs. But getting your animals enrolled in that type of program, getting estimated breeding values, and getting some connectivity to other producers that are in SIP is really, really critical for making the most appropriate, most accurate genetic conditions. We'll talk more about connectivity in just a second. Okay, so I talked about our RAM test model before, and now that kind of drove the selection in our Rambouillet flock. Since then, we've really got that flock, and then also our Dorpers transitioned to more of the NSIP type protocol and enrolled in that program. And so you see across the bottom here, this is just an example of an EBV profile from a buck that we produced a few years ago. And so there's some acronyms here I'll go through in case you're unfamiliar. WWT, weaning weight, post-weaning fat, post-weaning eye muscle depth, post-weaning worm egg count, and number of lambs weaned. And so there's different types of traits here. Weaning weight, post-weaning weight, those will be more growth-oriented. Generally, you would like those numbers to be higher than lower. Post-weaning fat and eye muscle depth. Again, this is a little bit subjective approach here. And so part of the discussion tonight, I think, is a bit on kind of Lee and I and everyone else's philosophies and approaches to genetic selection. And so I'll go ahead and say that I really prefer animals to have higher post-weaning fat and higher post-weaning eye muscle depth. And that's maybe more applicable to our breeds of sheep, but that higher fat value, I'm thinking when we think about females that are out in the pasture trying to stay in good body condition, I don't want them to have the genetics to be super lean. They're gonna have a tendency to be in too low of a condition score, and that's gonna affect a lot of their performance or production in various different types of traits. Now, if we had a terminal type breed, let's say Suffolk or something else along that lines, maybe we would not like to have a high fat EBV, because that's going to result in more retail trim when those lambs are harvested later on down the line. And so, breed by breed, production scenario by production scenario, this is going to differ. For me, I like high fat, high eye muscle, or high, more muscular type. Post-weaning worm egg count, we would like that to be lower. So what that number represents is a percentage. This animal here has the genetics to be 32% lower for fecal egg count compared to what was originally kind of that standard or breed average for the Dorper breed. And then number of lambs weaned, again, that's a percentage. This animal has the genetics to, or at least his daughters would be expected to wean 9.3% more lambs, again, than what was that baseline years ago. And so this is really a pretty good balanced EBV profile. Maybe it's not perfect. We ended up selling this ram back when he was a yearling. Probably never should have done that. There's a lot of good things to appreciate here. But I really want you to understand too, and this is an important takeaway, that just having EBVs or just having your animals enrolled in the National Sheep Improvement Program does not make them better in and of itself. That all it really does is it makes us more informed about them, and we can make more informed selection decisions. And from those EBVs, we can really match the right sheep to the right type of situation. And so I've got a table here across the bottom that I just pulled out the EBV profiles from 6 Rambouillet rams that we produced a year or two ago. And depending on the type of situation, the best buck in here might change. And so I look at like this ram right here, 7864, and I look across here. If this was a really dry situation where further west than even San Angelo, parasites weren't a problem, maybe they were had a parasite challenge 1 in every 5 years. The fact that he's positive for post-weaning fecal egg count, which is generally maybe not a great thing, we can be okay with that because he produces a lot of wool. He's a moderately sized sheep, doesn't have tremendous growth, at least when we look at this yearling weight breeding value. And so maybe that's the right type of sheep for that situation. Or if we do get into a little wetter area of the state and there are parasites and there's the forage available for animals to have a little higher reproductive potential, the 7843 might be the right sheep. He's got high growth potential, pretty good fleece in terms of pounds produced. He's negative for post-weaning fecal egg count. That's good. He's positive for number of lambs born. Maybe that's the right type of sheep in that situation. I'll tell you what, though, I kept one of these rams and bred him pretty hard this year. I'm really excited about, and it's this sheep down here, 7874. And so I mentioned earlier that in our Rambouillet flock, those ewes are probably a little too big, and so I'd really like to downsize them. So I look at this lower, weaning weight, lower post-weaning weight, really moderate yearling weight, and I'm not necessarily afraid of that at all. That's actually probably a good thing in this type of scenario. He's really low for fiber diameter. I don't have the breed averages across the bottom, but negative 1.1 is pretty good for the Rambouillet breed. He's a negative for grease fleece weight. Our Rambouillet sheep are already pretty good producers in terms of pounds of wool. And so, again, the fact that he's a negative, I'm okay with that. Part of the reason being is that when an animal consumes nutrition, it's got to be partitioned into various things. And I'd really like for these sheep to partition more of that nutrition towards immunity and ability to fight off parasites. So a negative 34% is great. And also the production of lamb, reproductive potential. So the fact that he's a positive 8.8% really fits the priority traits here. And again, we can live with this lower grease fleece weight, and we can live with the fact that maybe he's a little bit slower growing for the right type of situation. Again, all of this data allows us to make more informed decisions. It doesn't necessarily mean that all of these rams are better than a non-NSIP ram. It just makes that more of a mystery. That's all. Okay, so how do we get NSIP breeding values or EBVs in an extensive situation? And so I mentioned earlier that our flocks pretty much spend the entirety of their life out in the pasture on range. So how do we capture litter size? We do it with mid-gestation ultrasound. So if done at the appropriate time, we can really be pretty highly accurate in identifying the litter sizes of those females. Now, sure, is there some late gestation fetal loss that maybe confuses that just a little bit? That's a possibility. But we're going to live with that to get those animals into that type of situation. We capture, growth just like anyone else. We weigh lambs at weaning. We weigh lambs at a post-weaning time period. That's, 2, 3, 4 months later after weaning. We capture yearling weight. Usually those time periods also coincide with us doing something else to the animals. So we would obviously already be gathering them during weaning, so it doesn't add an extra work day to that type of situation. Usually during yearling, that's right around either shearing for our wool sheep, but even for our dorpers, we're starting to sort them out and make different management decisions. So they're already in the pen and they're already being captured one way or another. Capturing the body weight is not a lot of extra work. Parasite resistance for us can be a challenge. And the reason being is because we want to be able to get those fecal egg counts during that post-weaning time period. But the critical factor in that is that those lambs have to be exposed to a parasite challenge of some kind. We also run a fecal egg counting lab here in San Angelo, and too often I get fecal egg counts in from outside producers that want to do some genetic selection, but they weren't sure what the parasite load was out on the pasture, and they've gone through the trouble of collecting 100 fecal egg counts. They send them in, we look at those, and the average is 200. And in all reality, what that means is that those lambs were just not exposed to parasites, so we can't make informed genetic selection decisions based on it. And so it's really important that if this is a priority trait for you, that you're monitoring that worm load out on the pasture, collecting a handful of samples from a few of those animals to get an idea of where that contemporary group average is at. Then once it reaches at least 500 eggs per gram, but in all reality, it's better if it's up around 1,000 or 1,500 eggs per gram, collect everybody, and that's a much more informed data set than if it was too. For us, in those years when it's really dry and it's really too hot to get much of a parasite burden out on the pasture during that post-weaning time period, we have started to employ some artificial protocols. I think during Lee's presentation that you're going to hear in a second, he's going to describe a little bit more about how that works. But essentially, we're going to inoculate those animals with Barber's poleworm, get the fecal egg counts as needed for this type of evaluation. And then obviously, everything's born out in the pasture. We're not there to match up ewes and lambs, so we use DNA. We collect a tissue sample from all the lambs at marking, so that's 2 or 3 weeks of age. I got a picture of a TSU unit down here on the bottom right portion of the slide. You can send those in for genotyping, and that's how we conduct our pair. And so I'll go through that in just a second. So speaking of DNA testing, I do want to take a minute here and talk a little bit about genomic technology. So everything that we've discussed to this point so far, NSIP, estimated breeding values, that's quantitative genetic technology. Genomics is more molecular. And so that's where we're actually going to look at the DNA, look at the genome and the various gene mutations, and use that to make some type of genetic selection decisions. So some terms that you might be familiar with or may not be are genotyping. And so, in the cell of every animal, humans included, but in sheep, they've got an entire genomic profile, the entire copy of the genome of that animal. And that's going to be for sheep, 2.7 billion base pairs. And so when we do whole genome sequencing, that's where we would get all of those base pairs, basically get a reading or an output of that entire genomic sequence. Now, that's not necessarily cost effective when we think about trying to perform genetic selection. That's going to be too expensive. And so to reduce that cost and still get pretty valuable information, there's genotyping panels out there that only look at a limited number of those base pairs. So you might be familiar with something like a 50K panel or a 600K panel. And so Neogen has a 50K panel that's available for sheep and goats, for that matter, that genotypes 50,000 SNP or marker locations across that genome. So out of those 2.7 billion base pairs, it's going to look at what are the alleles at 50,000 of those spots. There's another product out there that you may be familiar with called Flock 54. That's from Superior Farms, actually offers that product. That is a 1,000 marker panel. So that's what we would call a low density panel, looking at very few number of markers. That 50K panel is a medium density, and then on up to the 600K or higher would be a higher density genotype. And so what can you do with that type of information? And so we, as I mentioned earlier, use genotyping and here at the station, and it's pretty popular in the U.S. sheep industry to use it to identify parentage. And really that's through kind of an exclusion method. And so just to explain that a little bit, just as a reminder, kind of a genetics 101, animals are going to inherit half of their genetic information from each of their parents. They're going to inherit one chromosome from their mother and one from their father, and so they have a pair of chromosomes. And so for each gene, you're going to have 2 copies of that gene. Or at each marker location, you would have 2 different alleles that are at that location, 2 either the same or different. And so when we look at lamb 1, or the lamb here, I've got, again, this is just an example of 4 different marker locations. And so the genotype for this lamb at marker location number 1 is AA. Again, this lamb would have inherited one of these alleles from its mother, one from its father. We don't know which one necessarily, but we know that one came from each of its parents. And so at marker location 1, lamb is AA. If we look at all of the potential sires that are of that lamb, we also look at that same location, and we realize that, okay, sire 1, he is an AG at that location. This lamb could have received this A from this ram. So sire 1 is still in the running to being the actual father of the lamb. Sire 3 is AA at that location. He too obviously is still in the running to be the sire of that lamb. But we look at sire 2, he is a GG. at that location, so he had to have passed on a G to his offspring. Lamb does not have a G. He's AA, so sire 2 is out of the running to be the actual sire. If we look at marker locations 2 and 3, the lamb is heterozygous at those locations. Actually could have received an A or a G from any of these rams. At marker location 3, this lamb could have received a C or a T from any of these rams. So that doesn't necessarily exclude any of these bucks from being the sire here. But if we look at location 4, where the lamb is a TT, and we look at sire 1, who is a CC, again, it's kind of the same situation. This ram had to have passed on C to his offspring. Our offspring does not have a C, so that excludes sire 1. So this method is used, if you were to use this method, you would identify that sire 3 is actually the ram that sired this lamb. And so it's a little more complicated than that, but this is essentially the way that genomics is used to identify parentage in the sheep industry and in other species as well. In general, for parentage, it's going to vary by companies and who's doing the analysis, but they're actually going to use probably 120 to 160 different marker locations to make sure that this is happening. Okay, what else can genomics be used for? Genomic information can absolutely help us determine the susceptibility of animals to certain diseases. And so if you're in the sheep industry, you've probably heard about scrapie. That's a common one. Ovine progressive pneumonia, rickets, dermatis paraxis. There's a few others. So these are just some examples. These are all all diseases that we have genomic marker tests for that can determine if an animal is more susceptible or more resistant. We're not determining, this is an important distinction, we're not determining if they have the disease. So if an animal is QQ at codon 171 of the prion protein gene, that does not mean that they have scrapie. It just means that they tend to be more susceptible to scrapie than one that would be RR at that codon. Now, there's also, we can determine if the animal is affected by certain genetic conditions. And so in this case, if the animal has a certain genotype, usually they have a particular genetic condition. And often these are not good. In some cases they can be, but generally these are deleterious or not good conditions. And so, for example, here, like spider lamb, I've got this table here on the right that explains that in this particular gene, FGFR3, that if a lamb is SS, means that it has 2 copies of a mutated version of this gene, that they're affected with the spider syndrome and often don't survive. Now, they can be a heterozygous carrier, meaning they have 1 copy of the mutated gene and 1 copy of the not mutated version. They're unaffected, but they are carrying that mutated copy. So if they were to be bred to another NS animal, there would actually be a 25% chance that the offspring would have the spider syndrome. And then obviously homozygous non-carrier would be unaffected and wouldn't have any offspring have spider syndrome regardless of who they're bred to. Okay, some limitations on genomic information. I'm going to try to speed up here a little bit because I know we've got another presentation to go. But in general, marker information can be really informative about conditions or disease susceptibility or particular traits that are controlled by one or a few number of genes. But when we get into many of our production traits like parasite resistance, reproduction, growth, etc., those traits are controlled by many genes. And so it really becomes complex when we try to look at particular genetic mutations or markers that are beneficial or negative for these types of traits. Often we'll find markers that are associated with these traits, but the influence on the phenotype is actually pretty low because there's so many other genes that are involved. And so I guess the takeaway here is that research is still at a situation. We are in the process of identifying markers that are beneficial for these traits, but I wouldn't expect really major silver bullet type gene tests to come out and really have high influence on these particular traits. Maybe the one exception to that rule that you may have heard of is the Borula mutation. There is a particular mutation of this BMPR1B gene that does really increase ovulation or ovulatory rate and can produce or cause females to produce an extra lamb or two in certain situations. But in all reality, that's a pretty rare situation. The other thing too is that there's really a crossbreed differences. So when we find markers that cause a particular phenotype in the end, or have a high influence on a particular phenotype in one particular breed, in many cases that doesn't necessarily translate to other breeds. Some cases it does, but not in all situations. So when we do genomic testing, we just have to keep these things in mind and as far as what our expectations are. Okay, we can have genomic information and quantitative information actually work in concert. And so here is a traditional EBV. What goes into an EBV includes pedigree relationship information, performance records, obviously the heritability information for whatever trait this EBV is going to be on. But we can actually include genomic information and increase the accuracy of these breeding values. And so how that works is that genomics can help us get a more accurate determination of an animal's relationship to others within its breed or others within the flock. So traditional pedigree style would be that an animal's 50% its mother, 50% its dam, 25% each of its grandparents, etc. When in all reality, they actually might be more related to animals, so there might be some inbreeding or etc. that goes on. And so genomic relationships actually are a more accurate representation of the relationships between individuals. And also what goes into a GEBV or genomic enhanced EBV are the combined effects of all the SNP that are being tested. And so I mentioned earlier that for production traits, the SNP usually have very small effects, but if we add those up, they can actually be a little bit more influential. So all that goes into a GEBV calculation along with performance records and other trait information. And really the benefit of genomic enhanced breeding values over traditional breeding values is they are more accurate earlier on in an animal's life. And that allows us to make more informed breeding decisions earlier on, not have to wait until a particular individual has lots of progeny records and is a little bit older in terms of the longevity standpoint. Now, for sheep industry, genomic enhanced breeding values are only available for the Katahdin breed through NSIP. There's a research project that's being led by a researcher at the University of Nebraska and in conjunction with the USDA that's working on getting genomic breeding values for some of the other breeds. And so hopefully in the next couple of years that will really expand. But as it stands, this is only really a thing that's for Katahdins. Okay, so how do we put all this information together and put it into practice? And so again, this is a bit subjective in my philosophy and my approach. I'm not necessarily saying it's right or wrong, and others might approach it a little bit different. But if I've got all this data and I'm trying to identify a stud buck or a keeper buck or keeper individuals, this is the way that I go about it. I first look at those EBV profiles and that quantitative information and think about what are the priority traits that I'm really trying to change. And so on this table here on the right, these are breeding values of Dorpers. And so the traits that I've just got here, obviously there's more, but weaning weight, post-weaning weight, eye muscle depth, fecal egg count, and number of lambs weaned. And fecal egg count and number of lambs weaned are the two traits that I really want to prioritize in my breeding program. So I'm going to look for individuals that are really superior in those traits. Maybe superior means top 25% of the breed, maybe it's top 10% of the breed, maybe it's top 5% of the breed. It depends on your type of situation and what works for you. But I'm looking for those animals that really excel in those two columns first. But I still want to make sure that I'm not picking an animal that is potentially not good in some of these other traits. Maybe they're not my priority, and maybe I'm okay with an animal that's just average for weaning weight or post-weaning weight or eye muscle depth, or maybe even a little bit less than average. But I want to avoid those individuals that are really on the extreme not so good end for these other three. And so I would like superior post-weaning fecal leg count, superior number of lambs weaned, and a balanced profile for the rest of that type of information. When I look in here and I'm looking at maybe some the bucks that stand out, here's a good example. This sheep here is negative 44% for post-weaning fecal leg count. Great. Positive 8% for number of lambs weaned, pretty good. But I look at its growth potential, and it's clearly the lowest out of everyone else in here. That's a red flag to me. Maybe that's not the right type of ram that should be kept for a stud. But I look on the other end of the spectrum and look at, so like this sheep right here, 22062, negative 13% post-weaning fecal leg count. Yeah, he's not the lowest, but that's still in the right direction. Positive 9, number of lambs weaned, that's pretty good. Pretty balanced and decent in these other traits. This is a sheep that is certainly in consideration for being stud buck. Kind of the same situation with this 22055. Okay, so I've looked at these breeding values. I've kind of picked out some of my favorites based off of that information. Then I'll look at the genomic information. I want to make sure that we're not selecting a ram that's carrying a potential marker or is a carrier for a disease that I really, or a condition or disease susceptibility that I really want to avoid in my flock. So that might help kind of sift those rams out. And then the last thing I'm going to do is absolutely some visual evaluation. I'm not necessarily one of those geneticists that believes that you have to look only at data and only at the paper, if you may, and not actually look at the animal. I do believe that still performing some visual evaluation is important and allows you to get a whole picture of the animal, because there's a couple reasons. There are some traits that common sense tells us probably has some genetic background, but we don't have an EBV for, or we don't have a marker for. Some of the examples like the parrot mouth, I got a picture down here, or really tight horns in our wool sheep. We want to avoid those types of things. And we don't have a good way of genetically testing for those situations yet. We're working on it, but we don't have that type of information yet. And we also want to make sure that we're not identifying animals that have something that's going to really prevent them from being productive or performing. So, I mean, obviously this picture here in the front legs of this lamb is on the extreme end, but we still want to look at structure, feet and leg conformation, and making sure that we're not breeding really, some problems into the flock, if you may. So when we're doing genetic evaluation, a visual evaluation, I really want to make sure that you're asking yourself, when I look at this animal and I'm selecting for this trait, is it something that's economically important? Is it going to affect my bottom line or the product that I end up selling in the end? And also, does it affect the ability of that animal to produce? So udder conformation, teat size, placement, those types of things are really important to look at. And we want to make sure that we're still, in some regards, selecting for animals that are preferable for those types of traits. Now, remember, we are making this decision based off of a phenotype. And phenotypes, we know, are based off of genetics and environment. And so there's some environmental noise in what we're seeing or what we're looking at. And so we just need to be really careful and make sure that when we think about our expectations and the ability to advance in a positive direction for these traits, that progress might not be made really fast because there's that noise in that types of situation. So if you want to ask some questions about that a little bit later, we can discuss that. Okay, I just have a couple more slides that I want to end with. I want to kind of bring this full circle and just take a second to discuss a research project, kind of a integrative research and extension project that we've got going on here at the research station that's using some of these technologies that I've described and some of these philosophies that I've described. And so this is a Dorper sire progeny test that we've just started hosting here in San Angelo. And really the goal of this test is to provide a genetic evaluation of rams. But the approach is we're bringing in outside rams from stakeholder flocks, and they're coming and they're breeding our AgriLife Dorper ewe flock, but then those rams are going to go home. And so we're actually doing very little measurement of the rams themselves. How we're making that genetic evaluation of them is based off of their progeny. And so all those lambs are going to be born. This is a fall lambing situation. They're going to be born and raised out in the pasture. And we're going to collect a whole suite of performance records on them. And we're going to have sire groups essentially. And so we'll be able to actually make some evaluations of those rams based off of their 20 or 30 offspring. Because remember, when an animal breeds, only passing along genetics, it's not passing along environment that actually might confound what you're looking at on that ram. So when we look at its offspring and make that look at those performance records, it's a pretty decent evaluation of his genetic potential. The key here is that these rams are not yet in NSIP. And so all of the data that we're collecting is going to go into that National Sheep Improvement Program. Hopefully going to get all of the flocks that are participating also enrolled in that program. And because each of them has provided a ram to this test, and there's offspring that are being reared from those rams in the same contemporary group, one of the big outcomes of this test is we're really promoting connectivity between those flocks. And what that means is that the breeding values that those newly enrolled flocks are going to get are actually going to be more accurate, because there's a bigger pool of data that those breeding values can be developed from. They're going to be connected not only to our flock, our San Angelo flock, but also everyone else that's participating. So that's really a big outcome of this project, and we're really excited about it. But kind of at a little more surface level, we're going to be able to provide genetic information about these rams in an extensive situation. Some of them did not come from big, large pasture ranch type scenarios. They came more from a farm flock situation. And we turned them out in 600 acres. We turned them out as a group and asked them to go breed those females. And so there's a lot of different things that are at play that are going to help make that successful. Ultimately, this is also going to allow us to identify some novel traits. One of the cool things that we're going to look at is we're going to harvest some of the male lambs from each of the sire groups and look at flavor profiles and kind of try to wade into that and sort of start looking at some potential for making some genetic selection in carcass quality, flavor, that type of stuff. Okay, so last slide, some take-home messages from what I described today. I know it went just a little bit long, but these technologies, I've really talked about them in the context of sheep, but they're available for goats too. EBVs, National Sheep Improvement Program, takes goat flocks, absolutely. Most of the genomic information or genomic technology I've discussed is available for goats too. I do want to make a point that advanced genetic selection is not necessarily for everyone. I don't want everyone to leave here tonight and say, you know what, I've got to get into NSIP. I've got to start generating EBVs. It's really for seed stock flocks, for those flocks or herds that are intending on producing breeding animals that are going to be sold out to other industry members. Commercial producers, while they may not need to enroll in NSIP because there is an expense with it, they might have too many numbers that it's really just not feasible. What's important for them is that they should buy genetics, rams and ewes and keepers that have this type of data, that have this type of information. It allows them to make a much more informed purchase that way. And so while I'm not encouraging necessarily commercial producers to enroll in the program, they still should be consumers of this type of technology. I really want to reiterate the point that I'm a big proponent of measuring animals in the environment that you expect them to perform in. And so, again, kind of thinking back to that extreme, we don't want to test something in a feedlot and then ask it to go and survive in a West Texas pasture where it only rains 15 inches a year. That's just not a great comparison. If you're starting out with NSIP, it's really best to not start at square one. And so what I mean by that is purchase animals that are already in the program, or get a ram that's got NSIP breeding values. It'll really allow you to have more accurate breeding values from the start. If you're just starting fresh with animals that don't have any pedigree information in the program, your EBVs for the first few years are not going to vary very much, and it's going to be a challenge. And lastly, The last kind of point that I want to encourage you is to find a mentor. If you're new in this, or even if you're not new in this, this type of technology can be complex, it can be complicated. So always having a sounding board or someone that you can go to, say, hey, what does this mean? Or should I be testing for this? Is really, really beneficial. So with that, I'll wrap up. I'll be happy to take some questions after Lee has a chance to go. I look forward to some discussion.
Thanks, Jake. And while we let Dr. Lee Wright get on sharing his screen, just a couple reminders. If you got any questions for Jake, put them in the chat so you don't forget. We'll get to those there at the end. And if you've just joined us, don't forget to put in where you're from so we can kind of keep track of that. And with that, I will turn it right over to Dr. Wright there at Virginia Tech.
All right, here we go. Can you guys hear me all right?
Yeah, we got you.
All right. Well, thank you, Adam. I appreciate that. Full disclosure, I'm not a doctor. Been doing this work at Virginia Tech for about 28 years now. So I got plenty of School of Hard Knocks behind me, I guess. We'll go with that. But it's a pleasure to be here with you guys tonight. A lot of what I'm going to talk about, there may be a little bit of overlap with some of what Jake discussed, especially when we're talking about the NSIP components. But let me see here. There we go. All right. So as far as making genetic improvement, and I think this is kind of from what Adam had told us, the things we wanted to talk about, basic principles of genetic improvement. Well, any trait that you want to measure, of course, I start talking and the train goes by. Excuse me. I apologize for that. Any trait of relevance that you want to be measured, you need to measure and record that to have a benchmark within your flock before you're going to make any progress with it. You really need to know where you stand to begin with. First thing you need to think about, what are your flock goals? From a management standpoint, first thing you need to think about is where you want these lambs to end up. What's your market component going to be? There's all kinds of different directions you can go with these lambs. I've got several things listed here from, seed stock and breeding stock producers. Are most of these lambs just going to be going to slaughter market? If so, what is your market? I noticed we had some people from out west. We've, just skimming through our list of folks here. It's not totally North Carolina or East Coast, trying to target that ethnic market that we can tap into. You've got some bigger feeder lambs operations that may be looking into trying to get bigger lambs and not trying to target that ethnic market. Grocery stores, restaurants, there's all kinds of different ways to go. But the one thing to remember here at the end of the day is, using genetic selection, it's going to help you meet those market needs. But again, you've got to know what your market is before you really start moving in the right direction. A statement down here at the bottom, this is just something I had pulled from another talk that I had given solely for Katahdin producers. And I realize we may not have all Katahdin producers here. But, even though Katahdins may not suit everyone, main thing you need to keep in mind is whatever breed you do have at home, use that breed for its intended purpose, and you're going to tend to be a lot more successful with it. In the case of Katahdins, they were developed to be a moderate framed, maternal meat sheep, did not require shearing or tail docking, kind of a low maintenance type of deal. So, be sure and focus on your breed selection when you're getting into the business. Basic management goals. I've got a few slides here that, really kind of bring it down to a basic level, but I think a lot of this information kind of holds true, no matter what we're doing, no matter where we're going. You know, you really need to focus on record keeping. You know, whether you're trying to keep those records to put into NSIP and generate those EBVs for your flock, or whether you're just using those records with a small flock, you know, you may only have 10 or 20 sheep. Records are really important to what you do on a day-to-day basis. Those records are going to help you know who's working for you. You know, anything from lambing records, inventory records, breeding, weights, from weaning weights to post-weaning weights, fecal egg counts, and again, culling records. Be sure you're keeping track of who's not performing for you. Anything from pocket notebooks to spreadsheets. And I've got, actually, I apologize. I just realized I didn't have that video link going. But, you know, a pocket notebook, I keep one of those during lambing season with me at all times. I've got a spreadsheet with, you know, each of those dams, who they were bred to, what breeding group they were in. So when those lambs hit the ground, you know, I've already got pedigree EBVs generated for those lambs when they hit the ground. I can keep a close track on how good my rams were doing in their breeding groups, kind of give me an indicator of whether I may have some problems with those. Anyway, records are really going to help you make better genetic selections, not just with EBVs, but all types of records in general. This is just a basic inventory sheet. You know, this just some of my animals. I've got a ear tag or ID number. You know, the status, are they active in the flock? Has she been culled? Did she die? You know, when was she born? What was her birth type? Some of that codon information, like Jake was talking about, you know, this is just a scrapey codon 171 information for each of those ewes. And of course, sires and dams, and just easy access to find and look up those registration numbers if you need them. If you're a seed stock producer, some of this information, even though it may be pretty basic, you can stick it in an Excel spreadsheet, have access to it anytime you need it. Lambing records, this, you know, that pocket notebook I was talking about, jotting notes down in it, leave plenty of space in there in case you've got problems with a ewe or a lamb, when you need to treat somebody for any particular reason. You know, go back and look at it and make sure whatever you treated that animal for, you can go back and look at it a few days later and say, hey, it worked, or hey, it didn't. I need to think about something else. As far as a lambing sheet, you know, this is just some basic information. You'll notice a rating score there. You know, sometimes I'll go through and in the comments section over here, if you look at it, this first ewe, of course, I've got keep on all of her lambs. She's a rock star in my performance system. So, you know, especially those females, I'm going to keep out of her. And I'm always looking for a good ram lamb out of her to use to cover more ewes with at a later date. We're going to get into some ram selection here shortly, but one thing to remember, you know, if you've got a really good ewe in your flock, chances are if you can get a good son out of her, he's going to pass a lot of those performance traits on that he's getting from his mother. So that's definitely something to keep an eye on. You know, culling decisions can be made at lambing time. You know, just taking a look at some of these notes over here, and these are just some things that I pulled just to show, you know, this 9019U, she's definitely on the cull list. You know, I had to pull a lamb. One of them she didn't clean, and one of them got chilled and she laid on it and it died. So, you know, out of a set of triplets, she's not faring too well. Maybe she's having 3 lambs, but she's certainly not doing a good job raising any of them. There's no need to keep her around. 9097, she may have a rating of 1. Okay, she's a good mother, but when you look at her history, she's had 3 years of singles. Is she really making you any money? Those are decisions you need to make. And by keeping good notes and good records on these animals, you know, it's just going to pay dividends for you in the long run. As I mentioned, we're going to get into ram selection. You know, here where I work at Virginia Tech and with the ram test that we've got here, we really focus a lot on these young rams, trying to make better performers, better studs for the future. And, you know, job one is finding that right ram that will fit into your program and do you some good, whether you're looking at EBVs, whether you're looking at test performance. I've got listed here most, but all in parentheses, genetic progress is going to be made through proper sire selection. So when you're looking or thinking about genetic selection and improving your genetics with a ram, you know, you got to think about that ram is pretty much going to be half of your lamb crop. Okay, so you want to buy from a reputable breeder. Try and find those individuals that are in the business of raising good rams. Find folks that are keeping good records and really, you know, put a vested interest into their animals to make sure that they're selling good animals to everyone else. Performance ram test. Jake mentioned their Dorper test. That sounds like an awesome thing. The ram test we've got here at the Southwest Ag Research Center, you know, we've been going strong since 2012. And, you know, evaluating these rams for post-weaning growth and fecal egg count and parasite resistance, you know, I think we're onto a good thing here. And I think a lot of our customers and consigners that have come to this test and our sale have been well pleased with their outcomes. Again, you know, the daughters of these rams are going to have characteristics of that ram's mother. So definitely want to take a look and see those records. And, you know, heritable traits, all these things that we talk about that you can find these values for in NSIP, to a certain extent, you can go back to about all of these traits and that ram is going to pass those traits on. So this is not the greatest picture in the world, but, you know, when you start talking about ram selection, how do you choose one? You know, you walk up, guy's got a couple of rams for sale. You know, which one of these do you want? One on the right, one on the left. Well, first thing I'd ask, just looking at them, you know, I want to see some records behind him. If he doesn't have any records and this is all I've got to go on, then, you know, probably you're going to take the ram on the right. He's a little bit bigger, a little heavier boned, a little more masculine looking. He's got more size to him. His frame is a little bit better. You know, not getting into a judging class here, but, you know, and some folks, hey, you want some color? You can call him Lover Boy. He's got a nice little heart imprint there on his front shoulder. So, you know, just based on looks, maybe we pick the ram on the right. But, you know, when you dig a little bit deeper and you find those records, you know, the ram on the right, he's 2 years old. He was a single birth type. He was predominantly fed grain in first year of his life. And turns out he's in the bottom 5% of the breed for parasite resistance. Ram on the left, well, that guy's only 10 months old. He's still got some growing to do. You know, he was a triplet when he was born, and he was raised on pasture the whole first year of his life. And turns out he's in the top 5% of the breed for parasite resistance. So unless you have records to show that, you're kind of going blind ram shopping. And I've just got a couple of little quick examples here to talk about too for selecting rams. Again, you walk in looking for a ram. Okay, so you got 2 yearling rams. You know they're both the same age. One of them's 140 pounds, one's 160. Okay, so really all you've got to go on is one had a little bit more to eat than the other one, maybe. But maybe it looks a little better. Maybe it's a similar situation to that last picture we looked at. Well, if you start to dig a little bit deeper, you know, each one of these rams turns out they had 25 progeny last year. They used them to breed some ewes too, and say each one of them has 25 lambs. Well, if you look at Ram A, who's the smaller ram, it turns out his 25 progeny averaged 50 pounds. Ram B, the bigger ram, his offspring didn't average but 40 pounds. So just off the bat, Ram A is already making you more money if you're selling lambs by the pound. So if you want to continue to dig a little bit deeper, we've still got that last scenario where Ram A's progeny are 50 pounds and Ram B's are 40. But you know, you need to ask a few questions. Turns out, Ram B was only mated to young ewes and Ram A was mated to older ewes. Well, right off the bat, Ram A being bred to those older ewes, they're going to be producing more milk. So at weaning time at 60 days, those lambs would be expected to be heavier than Ram B's. Because if he was only mated to younger ewes, their milk production is not going to be near what an older ewe is. So they're already at a disadvantage. What if Ram A's lambs on average were 20 days older? That's definitely going to account for some more growth. What if Ram A only had 3 lambs in his group and Ram B had 30 lambs? Law averages is kind of going against Ram A at that point. So, these are just questions you need to ask and things you need to find out. So when you're buying from a producer or a breeder, seed stock breeder, they should be able to answer these questions for you. They should be able to pull their records and answer anything you have. So what do we do with all this information that we've asked and we've got, how do we combine it and figure out where to go with it? Well, Jake had touched on some of this, so I'll skip through some of these NSIP slides fairly quickly, but one thing to consider, you have all that information, you've got your performance records, you've got your pedigree, so sires, dams, grandsires, grand dams, you've got your full sibs, half sibs. You may even have progeny data on those animals. And by looking at EBVs and using the NSIP program, it's going to make those adjustments for the age, the sex, the age of the dam, what the litter size is. All of that's going to be taken into consideration. So when you have those EBVs to look at, it helps you sort that information out in a much cleaner way. This is just a list of some of the important EBVs that are out there. I know Jake covered most of these in his talk, so we're just going to skip on over this slide. But I will say here, if you go to the NSIP website for February of 2024, here's the percentile rankings for the thousands of lambs that are entered into that system that have data behind them. And with those percentile rankings, basically, when you look at this just gives you a gauge. If you look at the 50th percentile there in bold across the middle, those are a lot of about where the average falls for those breeds. You can look at the top 1% or you can look at the bottom 10%. So when you're looking at EBVs on animals, if you're shopping for rams, if you're shopping for ewes, this is a good resource to have. There's a searchable database on this NSIP website as well, where if you've got the IDs of those animals, you can go in and sift through some of their pedigree information. You can find animals that way. And again, if you've got a listing of potential rams that you're buying from a seed stock producer and he's furnishing you with EBVs, you can use this percentile ranking just to see how those animals stack up within the breed. So how do I personally use NSIP? From a growth standpoint, my kind of school of thought with weaning weights and post-weaning weights both, I kind of push the limits with those, just, see how my system handles it. You can increase those numbers and you can get better growth rates out of them, but there's a nutritional component to that as well. So, you've got to remember that if you're selecting for bigger growth or much higher growth rates in those animals, they do need nutrition to meet some of those expectations. Again, going back to your market, what's your market? When do those lambs need to be ready? Depending on what your lambing season is and what kind of target market you have and how big those lambs need to be, you can use some of these growth EBVs to kind of push those lambs along and get them ready for when they need to go somewhere a little bit faster. One thing to think about, from an ethnic market standpoint, that's a moving target. A lot of those holidays get about 10 days earlier every year. So if your lambing season stays the same, some of those holidays that you might be trying to target for a marketing avenue, if they're getting earlier, you're going to have to get weight on those lambs a lot faster. So you can use some of these growth EBVs to push those lambs along and get them ready a little quicker. Higher growth EBVs can give you some management options with those lambs, whether they're on grass or feed. But one thing to remember, you know, if you get too high with these growth numbers, they can adversely affect the mature ewe size if you're raising your own replacements. So, you know, if you're pushing your growth limits on the sires that you're selecting to bring home and use into your ewe flock, but you're keeping your own replacements, those ewes are going to naturally be bigger. They're going to be a little framier, and they're going to require some more groceries. Which leads us straight over to the maternal side. If you are keeping your own replacements, you know, balancing those numbers, balancing those EBVs, whether it's growth numbers, whether it's maternal numbers, you want to keep a good balance in there. Don't get too far out of whack with any single trait selection on your ewe flock. That's just kind of how I've approached it over the years, and it's really paid off, I think. So, you know, there's a term that was coined by Roxanne Newton down in Georgia called superior average. So if you go back to that last sheet we talked about, or that percentile ranking report, and you look at what average is, and you go a little bit better, but you don't push those thresholds at the high end, a lot of times, more than not, you're going to have a much better flock at the end of the day. You know, as far as I'm concerned with ewes and those maternal traits, I want to have a ewe that's going to be a consistent twinner. She's going to raise them. So if I look at those number of lambs born and number of lambs weaned categories on that percentile ranking, I'm going to want to hold somewhere between that 10% and 20% range. If you get much higher over that 20% range, you're really starting to, you know, you're going to, especially on the number of lambs born, that's probably an indicator that she's going to be having a lot of triplets. So unless you want triplets, don't push those numbers too hard. The maternal weaning weight, that's kind of the old maternal milk number. If any of you have been looking at the system for a while, or if you're familiar with cattle EPDs, you know, that maternal weaning weight number, I like to keep it right around 1 plus or minus a few. You know, I'll let it drop to a 0.5, but I don't usually push it much over a 1.25. If I go much higher than that, you know, a lot of times those girls aren't going to meet your expectation for that unless they're getting heavy supplementation. And if you're in a pasture-based system, or if you're only supplementing a small amount of grain, you know, you may run into some mastitis issues. Sometimes you could just push that number a little bit too hard as well. As far as the index value, it's the hair index in Katahdins. That's kind of a combination of the maternal weaning weight, number of lambs born, number of lambs weaned. It takes all those maternal numbers into consideration, and it gives you a single value on that index to kind of give you an idea of where she stands from a maternal standpoint. There again, if you push that number too high, again, knowing the number of lambs born and number of lambs weaned are factored into that, that could be an indicator of more lambs hitting the ground, which means you're going to need more milk to keep them growing. But if triplets is not a goal, then you really need to be careful how you look at those numbers and how you use them. If you're in a grass-based system, you definitely want to take into consideration your fecal egg count values, and we'll talk more about those later on. But again, if you look at that percentile ranking that was up there, that -20 is somewhere around the middle of the pack, that 50th percentile, and the -70 number is actually pushing up close to the 90th percentile. So you're getting really close to the top 10% of the animals in the breed for parasite resistance. But anywhere in this range, I think you're going to have a good animal in a pasture-based system. The more negative those fecal egg count values may be, we have seen in some of our research that it could be an indicator of just better immune function altogether. And we'll talk a little bit about that shortly as well. So why is post-weaning growth critical? You know, what you really need to think about from a post-weaning standpoint, these animals, once you wean them off of mama, they're going through a stress period. Growth and feed efficiency for these market lambs is going to be key. To meet those marketing opportunities that you have, you know, whatever your market may be, the growth of those lambs is the really most important part during this post-weaning phase. Structural and reproductive development, the growth stage leading up to puberty, that's exactly where these animals are right now. So that is a critical stage in life for them. Nutrition, proper nutrition and development is going to lead to breeding efficiency in both the males and the females. So looking at these post-weaning numbers is really key for any breeding program. Early life growth and health for these lambs is going to lead to reproductive longevity in your flock. So the better care you take care of them during this stage in life, it's going to pay dividends for you, especially with those replacement ewes you're thinking about keeping, or maybe that replacement ram that's in there that popped up that you think might suit your program very well. So as far as measuring this post-weaning growth, key thing you need to remember here, weaning is a big stress on these lambs. You're taking mom away, they're getting out of what their daily routine is that they've known for their entire life. So it's a huge stress factor. If their immune system is not fully developed at this point, which it hasn't, stress is going to induce a lot of other problems. Where are they? What kind of diseases are they exposed to? Is there coccidia around? Are there parasites around? If they're on pasture, or if they're anywhere around a loafing lot or a dry lot or watering trough, I can about guarantee you they're exposed to coccidia. If they're on pasture, they're exposed to parasites, and they're always going to be exposed to these clostridial diseases as well. So be sure you're vaccinating these lambs with CD&T at a minimum to help avoid some catastrophes during this post-weaning phase. Just talk a little bit about the RAM test here, which, really kind of hones in and focuses on this post-weaning growth and parasites. These are two really important things for ram selection as far as I'm concerned. The rams that we bring in here, if you're not familiar with the program, these lambs are born anywhere from January 15th to March 15th. They come in here around June 1, right after Memorial Day is typically delivery day for these rams. So they've got to be pre-weaned, they've got to be pre-vaccinated. There is a weight minimum for them to come in and participate in this test. Once they get here, we give them all 3 dewormers, 3 classes of dewormers, try and clean their system out before they get here, and they go on dry lot for a couple of weeks, just to kind of acclimate to the feed that we'll be giving them. Make sure we don't have any health problems going on. Just settle them down and kind of work them into a routine here at the station before we put them out on their pastures. After that acclimation period, once they go out on their test paddocks, we actually dose each of those rams with about 5,000 worm larvae. So, when they go out on test, we know they're getting a dose of Homunculus worms, and we know that once they're out there on pasture grazing, they're going to be picking them up even more on a daily basis. So from a growth standpoint, we're monitoring the average daily gain on these animals. I've got a couple of slides coming up here that just show some results from a particular year that I just picked at random that I had some information for. For this particular year that I selected, the average for that test group over a 70-day period, there were 117 rams on test, and the average of the group was 0.37 pounds. And just to give you an idea of what their daily consumption is, they're on a fescue-based pasture. It's got some orchard grass, some clover in it. It's kind of a typical pasture setting here in Southwest Virginia. We do supplement them with 2.5% of their body weight and a 16% crude protein pellet. It's got a mineral package in it. It's pretty high fiber. It's nothing fancy. But we know they're getting a little bit of extra protein each day because sometimes our pastures, if we have a dry year, those pastures are going to get crunchy and, maybe not be quite as good a quality. And we've had some years that have just been excessively wet, and we know our pasture quality has not been good then because it's been predominantly water. So, they get a little bit of extra protein bump from us from that daily supplement, but they pretty much come in at feeding time. We dump it in the trough. It's gone within 5 or 10 minutes, and they're back out on the pasture grazing every day. Those rams are split into 3 contemporary groups, and it's based on weight and age. So when you're selecting rams, utilizing this test data, you're going to double your chance of success here by using NSIP or some of these previously tested animals. So this is just kind of a breakdown of how, the test ran that year. I told you it was 0.37 pounds was the average for the average daily gain. So, if you look down here at rams from NSIP or previously tested sires, these are rams that have already got information behind them. They have EBVs or they're from a sire that has previously been through the test. So he's got some data behind him. And those producers that are bringing those rams here and consigning them, they've got some background information. And if you look at the bottom line where rams are from non-tested or non-NSIP flocks, the total rams, we had a few more NSIP rams than we did non-NSIP or non-tested rams. But when you look at, who performed better on test, who was higher than the average daily gain, 61% of those rams that were on test with NSIP or previous test data behind them, 61% of those rams did better than average. And those non-tested rams or non-NSIP sires that had no information behind them, only 35% of those tested better than average for the average daily gain that year. This slide is basically just telling you what we talked about with the numbers on the previous slide. So the blue dots are the previous tested rams from previously tested sires or NSIP sires, and the red dots are from the non-tested non-NSIP sires. So the blue dots definitely way heavier to the left end of this graph, which is higher average daily gains. So by using some of this test data or using EBVs, you can incrementally make progress with that growth data by using these rams. So take home points on your post-weaning growth. Again, your market strategy, whatever that may be and whatever your market may be, the higher growth values can help you meet those targets and get those weights faster. But you can't forget that nutrition component if you're selecting for higher growth. Faster growth genetics may not perform as expected in a totally grass-based system. So that's one thing I really want to drive home. If you're an all grass or all forage based system and that's how you want to stay, that's certainly fine with that. I have no problem with that whatsoever, but forage quality and forage quantity are going to be crucial for you to meet the expectations of what these rams are that maybe have some of these higher growth numbers or growth potential by using EBVs. Pitfalls on post-weaning growth EBVs and trying to go higher with them or using high growth rams from test data. Again, you want to think about your replacement breeding animals. These are going to be your ewes that you're bringing home or keeping at home and putting in your flock. You're probably going to have some larger framed ewes if you're not careful and if you push that threshold too hard. Meat market lambs and breeding stock are two totally different animals. Okay, so if you're breeding those lambs with a high growth sire, maybe you're not going to keep any replacements out of her. Maybe you're only looking for that ram to meet that market lamb goal, and you've got another ram that you're putting on ewes to keep replacements out of, and that's totally fine. But if you're only shopping for that one ram, or you want to keep a lot of progeny out of this one ram, these are things to definitely consider. That frame size and feed efficiency is going to hit you pretty hard if you're not careful. And down here at the bottom, how much are you going to have to feed that big ewe to maintain her and her lambs during your next lambing season? Because she's going to consume a pile of groceries. So we're going to shift gears just a little bit. We're going to talk about the fecal egg count side of things and parasite resistance. And again, goes straight back into ram selection 101 here. This graph shows kind of a typical trend of what we see from most of these rams that we have here on test. So that June 29th, that's going to be when they go on test and we dose them with that 5,000 worm larva I was talking about. So the first 2 weeks of that, you're really not going to have a whole lot of increase in that fecal egg count. And that's basically because, we're giving them third stage barber pole worm. Those worm larvae still need to develop, hatch out into adults, attach themselves to the inside of that gut and start sucking the blood of these animals for us to see that parasite challenge. So the first couple of weeks is not a big deal. Don't expect to see a whole lot of change. But that next 2-week interval, after those worm larvae have hatched out and become adults and started to feed, that's when we're going to see that challenge, and we're going to see a lot more of those eggs getting cranked out in the fecal matter because of those adult worms being reproductive in their system. So what we want to see with these rams is when they hit that spike and they go up as high as they're going to, this one is just happened to be a 1200 where it spiked. Well, then that ram, its immune system kicks in. It says, hey, we got a problem here. We need to do something about it. That animal recognizes that it's parasitized. Its sort of immune system kicks into gear and starts to get rid of some of those worms on its own. And he sort of comes back down, plateaus, and he's fine. He doesn't really require a deworming. He just managed it all by himself. This just kind of gives you an idea of some of the things we've seen. That last slide was kind of the average of what we see over time. Here we've got 3 different rams. Ram A in the green is very typical to what our average is. He's got that sharp spike that goes up, that fecal egg count gets high. He comes back down on his own, didn't have to be dewormed. He managed it. Okay, he's got some resistance or resilience per se. He managed it on his own. He's going to do fine. He goes through the duration of the test at a lower fecal egg count value. His immune system is kicked into gear and works great. The blue line ram, same trend. He didn't spike near as high, and he almost dropped back down closer to 0 and stayed at the bottom. This ram has got excellent parasite resistance from this is what we're looking for with our test. These are the rams that are going to offer you a great package of parasite resistance genetics to hopefully pass on to their progeny. And if you can combine that with some better growth, looking at the other side of the test data, you're looking at a really nice ram to take home and use as your next herd sire. The ram in red, Ram C, that dude spiked and he never came back down. His immune system never kicked in. This ram is very susceptible to parasites. He just kept climbing. He's eventually going to need to be dewormed, and he offers you nothing in the realm of parasite resistance whatsoever. So let's go back to this percentile ranking as we talk about this. And I know Dr. Weaver is on here with us, so I'll go ahead and say thank you for some slides I was able to pull from one of his presentations in years past. We'll even give Dr. Bowdridge a little bit of credit with this West Virginia University logo slapped up here. But over the years and working with Dr. Weaver and Dr. Bowdridge and Dr. Greiner, a lot of work from Virginia Tech, West Virginia, and NC State has been done here at our site in Southwest Virginia. So I always want to pay a little bit of tribute to these guys, because if it wasn't for them, we wouldn't be where we are. But, looking back at this percentile ranking, this is just from 2019. So these next few slides are basically pulling some of this test data that we saw, or pulling some of the other research that we've done here from a fecal egg count standpoint and looking at that. So, you pick out the very high and the very low. The best animals on this percentile ranking in the Katahdin breed are -100 on their weaning and post-weaning fecal egg count EBVs. On the opposite end of the spectrum, the worst animal in the bunch was a plus 1,832. That's probably at least the red line on the last graph, or maybe worse. So when you look at the normal distribution of what that percentile ranking is telling you for the fecal egg count EBV, if you look back at it, that breed average or 50th percentile was right around -51. So you definitely want to be shopping for anything between that -51 and the -100. Okay. Anything that's not at least average for these fecal egg count EBVs and greater, then, they may do more harm than good, depending on what your euflock base is. So again, what we want to do is try and find those lambs that are closer to that negative 100. Those are few and far between, I'll tell you that. Negative 90 to negative 100 are pretty rare, but there are some out there. But, just so you know, between that blue line at that negative 51 to that negative 90, those animals in there do offer some benefits in pasture settings. So what have we done here at the research farm besides the ram test? Basically we took some extremely high sires for fecal egg count EBVs and extremely low sires for fecal egg count EBVs, and we mated them to our Katahdin euflock. So this just gives you the idea or the breakdown of that post-weaning fecal egg count EBV for the 4 low sires we used and the 4 high sires that we used. So we tried to find as low as we could, and we really had to shop around to find the high side. A lot of people, they either don't keep them if they know what their values are, or they die. So anyway, we brought those animals in here. So just to give you an idea of what the average is for those low sires is negative 87. And the average for the high sires is a plus 335. So we were really going to push them to the limits. So what were our lamb EBVs? What did they look like after that progeny hit the ground? Well, of course, we've got the EBVs from the sires and the dams. So we've got the pedigree information, and then we collect those fecal egg counts here on site. We submit that data into NSIP as well. And this just gives you the breakdown of what the average of the low sires, the average of the high sires were. And on the right, for the weaning fecal egg count at the top and the post-weaning fecal egg count at the bottom, it gives you the breakdown of what the averages were per sire. So each of their progeny, how they performed. So when we look at weaning time here, we see this blue line pretty much looks like what we wanted to see in our ram test, if you'll recall. Weaning, they're approaching that stress period. They're getting older, they're starting to eat more grass, they may start picking at a little bit of creep. They're looking for other avenues of nutrition other than just mother. So once weaning time hits, you can see that spike in that fecal egg count. A lot of that's due to stress. They may have worms there, but they're still getting the extra protein bump from mom. They're still in a comfort zone. They're still happy. There's not been a drastic change in their life. But once weaning hits, you're going to see that spike in that fecal egg count sometime within the next 2 to 3 weeks, more times than not. So how did those lambs react? Well, when you look at, on average, those low fecal egg count lambs, they had their spike during that stress period. They adapted to it. Their immune system kicked in. They leveled back out. Fecal egg counts dropped, and they did a little bit better. The high fecal egg count lambs, on average, they just kept climbing, which is really what we anticipated to see. This graph just gives you the breakdown of the 4 sires and the averages of how each of their progeny groups performed. So, as you can see in the low side, there was one that went up, down, and back up again, that light blue line. So, a little later, his lambs were reinfected, got challenged again. Maybe they spiked again. And some of these lambs, we probably had to deworm some of them, but, the lambs that got dewormed sort of got kicked out and were factored into a different category at that point. But these lambs that went through this entire test without having to be dewormed are what you're looking at here. The high fecal egg count sires down here in the bottom right, 3 out of 4 of those, they just kept climbing. One of those rams, his lambs sort of did a little bit better and maintained, but for the most part, they kept going up. So as we've looked at this fecal egg count EBV over the years, and we've really been focused in on parasite resistance and how to control barber pole worm, this particular year sort of had us scratching our heads thinking, maybe there's more to this fecal egg count EBV than just parasite resistance. When you look at year 1 of this study, This is the death loss. You know, the blue bar are the low fecal egg count sire progeny. The orange bar are the high fecal egg count progeny. You know, in year one, right around weaning time, we ended up with a 30% death loss in these high fecal egg count lambs. But it wasn't due to parasites. It was due to a type A Clostridial outbreak we had unexpectedly. But it turns out, you know, the majority of those lambs and that 30% death loss from high fecal egg count sires, you know, came from a whole nother disease that we had not been looking for at all. Even in year two, when we vaccinated for that type A Clostridial along with our CD&T shots, you know, we still almost had a 5% higher death loss, even with a vaccine, to those lambs in year two over the low fecal egg count rams. And this is just some other information we collected as far as foot scald issues that we had in the spring. Typically, we'll see that pop up here when it gets wet. Fescue starts to grow. Any of you that have fescue-based pastures, you know that grass can be, if you rub your fingers backwards on a blade of fescue, you know, it feels like little shards of glass. And a lot of times that'll cause some wounds to open up in between those toes, and we'll tend to get some more foot scald issues show up that time of year. And this just gives a breakdown of the foot treatments by sire. And, you know, 10% less foot problems with the low fecal egg count lambs than the high fecal egg count lambs. It wasn't near as drastic as what that death loss was in year one in that previous slide. But again, it still shows that there's maybe some benefits to this fecal egg count EBV that we haven't quite tapped into yet. And that's where a lot of our research has taken us, you know, in a different direction now, but still focusing on that number. So in conclusion and wrapping things up here, you know, rams are going to be half your lamb crop. So, you know, what is that ram going to be worth to you? You know, whether you've got 10 ewes, 20 ewes at home, whether you've got 100, whether you've got 1,000, you know, those rams that you put in with those ewes that you have are still going to be half that lamb crop. So it's going to be worth a significant or make a significant impact to your bottom line. The genetics you're getting from these rams, you know, we talked about the growth component, we talked about parasite resistance, touched a little bit on the maternal traits that they can pass along. So, you know, they can have a huge influence on where you go and what kind of genetic selection and genetic progress that you want to make within your flock. Numbers don't lie and looks can be deceiving. So, you know, we saw that pretty evident in a couple of slides here that we've looked at tonight. Always look at the big picture. You know, if you want to improve your flock, you've got to know where your weak points are, and you've got to be able to identify the animals that can help move that chart, take you to the next level, make improvements on what you need to do to fit that market and your goals that you've looked at and that you have identified that you need to pay close attention to. And again, genetic progress can be made exponentially faster with a ram than it can a ewe. That ram is going to have so many more progeny hit the ground than that ewe having a single or twins or triplets each year. You know, the ram impact, you know, he's going to be putting 50 or 60 progeny or more on the ground. So, you know, as far as keeping those replacements, you can make progress a lot faster. And that's all I have got, Adam.
Well, appreciate that, Lee. A good presentation. Before we get started with Q&A, if anybody does, we do have a evaluation. I just put that link into the chat. It's a Google form. If you don't mind just taking a couple minutes and filling that out, when you get a chance this evening, before you forget about it, that really helps us kind of evaluate our programs so we can always improve those. But with that, we will take any questions for Lee or Jake. And feel free to unmute or put them in the chat. And this webinar will be recorded. Well, it has been recorded, but we'll take that and put it on YouTube, where Dr. Weaver has posted the previous 3. So you can find those there, and I think we've been sending those out through the Eventbrite emails that you signed up with. So we'll send that back out, and we'll also send the evaluation link through that. And you can also find the previous year's webinars that we've had on that YouTube channel as well. And we'll wait just another moment or two for any questions. So we'd just like to appreciate Jake and Lee for coming on. So we have a question. When is Lee's ram test auction?
It will be September the 20th this year, if I'm not mistaken.
And feel free to reach out to us. We can get that information if you think of it later on or need that again. But again, we just want to thank everybody for participating in the webinars this year. Stay tuned for announcements for next year's webinars, as well as some other events that we'll be having until then. But if nobody else has any questions, again, appreciate it. Take time to fill out the evaluation out, and we'll see you. Oh, we got one more here. Do you have any recommended resources for either one of you?
I just attached our website, so you guys feel free to click on that and head to it. You know, my email, phone contact, everything's on that website. You know, I'm happy to talk sheep anytime.
Yeah, I'll say the same. I'll put my email here in the chat in just a second, but I would some of the NSIP stuff. NSIP.org has a great. That's kind of the the website for the program, and they have a lot of resources on there. There's a lot of PDFs, there's a lot of videos, there's lots of different things that you can take some time to kind of peruse through, and it's really informative. So I would say nsip.org is a great spot if you're looking for more information about that particular program.
And Dr. Weaver said he will post a list of the local sheep and goat sales for 2024 later on our NT State Extension's website. So again, thank everybody for coming on tonight, and we will see you at some point, and appreciate those who've joined us all 4 times, and we'll hope to see y'all get on here next year. We actually have one more question if y'all got a second. We have, says, I have 30 ewes in my flock. High performance rams can be expensive. Any advice?
What's it worth? Genetic progress comes at a cost, right, Jake?
That's right.
So yeah, it's you know, it's one of those things that in my opinion, you know, if you follow sales, if you're talking about being expensive from an auction, whether it be a online auction, whether it be the ram test auction here, I know sometimes the prices of our rams, I scratch my head over them. But you know, it's still a good way to identify producers that have quality animals, and maybe you can back up with that producer or consigner to one of these sales that has sold a really high dollar animal. You know, maybe they've still got some at home. Maybe they've got a half brother, you know, and you can buy them a little less expensive off the farm. So, you know, there's some other avenues to go, but again, if you're really needing to make genetic improvements in one trait or another, if you can identify those animals that can do that for you, you know, I'm not going to say cost is no object, but you know, talk to folks that are in the business.
Yeah, I'd agree. And you could also maybe explore some opportunities to, partner on a ram with someone. If you only need him for, part of the year or your breeding season, and he could go somewhere else to your partner, and that would be one way to potentially reduce that cost. Maybe look for aged rams. I'm not going to say that that's always the best, but you might be able to find a certain situation where you've got a ram that's been used by somebody for 3 or 4 years and they're ready to move on. He's got lots of progeny. Maybe they're in that. Hopefully they're in NSIP. Or EVVs on that buck would be pretty accurate at that point. That might be a good option. And you don't necessarily have to have a world beater for every trait. I mean, again, I think both Lee and I touched on the importance of balance in terms of genetic potential and a balanced across the board for various traits, and making incremental progress, but making sure that you're not going backwards or drastically backwards for something else at the same time. So again, usually the rams that bring the highest value at certain sales are those that are really superior in certain traits. But if you kind of are willing to back down to maybe that top 30% or even top half of the breed for some traits, you can usually find a much more affordable genetics that way too. So those are some options.
And, prime example, just to reiterate that, Jake, we sell the top 50% of the rams here on test. So we draw the line in the middle. They've got to pass the test to start with, but we only sell the top 50%. Even though you're not buying the top 1, 2, or 5 rams in the sale based on how we rank them, that's still the top 50 rams out of 100 that are here on test. So there's still some good rams that are sold at decent prices.
Is there any other questions anybody has this evening? Well, if not, everybody have a good evening, and we appreciate you joining us tonight for the final webinar for the winter webinar series.
Thank you all very much.
Yeah,
Really appreciate
thank you guys.